Hello, everyone. It's really my pleasure to host Dr. Hadjantonakis from Memorial Sloan Kettering Cancer Center today. So her work is really truly beautiful. It involves quantitative microscopy. So Dr. Hadjantonakis grew up in England and then came to Canada for a postdoc and has been at MSKCC, I think, from the start of her career, where she's now the chair of developmental biology. But it's really a beautiful trajectory of how all of the pieces are in place to answer really some important and complex questions of mouse embryology. So when the early work from her lab started developing these beautiful fluorescent reporters that would mark the events of the very first cell fate decision that happens in the mouse embryo between what becomes the embryo and what becomes the extra embryonic tissue. And after developing these reporters, she developed wonderful systems for staging these embryos at various stages of the development. Do remember that the mouse embryo is really, really tiny and has very few cells at this point. And again, pioneered the use of keeping them alive in culture outside of the uterus, and then also developing the techniques for quantitative imaging. And in my mind, that is really the first single cell technique, right? If you can follow one cell by fluorescence and quantitatively image how much there is, I think that's how all of this single cell genomic craziness that goes on now started. And then her lab has discovered important signaling pathways that control the allotment of these cells between the kinds of tissues. She's found that amazingly, both embryonic and extraembryonic cells actually contribute to the gut endoderm. And this she found by, again, microscopic techniques a while back. And more recently, she has used in collaboration with Dana Pierre's lab, how the segregation actually leads and how that ends up in the embryo contributing to the endoderm. She's gone on to mutate and get rid of various transcription factors, shown that SOX17 and NEOMES are important for various kinds of patterning that happened in the early embryo. So I think it's really a synthesis with pioneering work, starting from how to develop models, starting from difficult techniques, and getting really beautiful quantitative information from really gorgeous images that we will be treated to see today, and in ending up with something we do in my lab, which is just transcriptomics, just measure, but not observe. So I really look forward to your talk, and thank you for coming here.
Thanks, Rupa, for the great introduction. So what I thought I'd talk about today is I realized that I'm going to have a colloquium and meet the students afterwards. I gave the students a paper to read, and they came up with some really, I think, insightful questions that actually I think part of them I'm going to answer today. So what I'm going to do today, as Rupa alluded to, we use high-resolution imaging techniques. We use the mouse embryo and stem cell models to understand mammalian development. What I thought I'd talk about today is something unpublished. It's really very much a work in progress. And as Rupa said, there's all this single cell craziness happening at the moment. And what I'm actually going to tell you today is our current attempts to perform spatial transcriptomics and actually spatial epigenetics. So really trying to take single cell genomic data and annotating it spatially, but not just spatially in 2D as many of the commercial platforms such as 10x's Visium are doing, but doing it in 3 dimensions and at single cell resolution. And I think many of you will appreciate that the Visium platform, which is one of the prominent commercial platforms, does not give you single cell resolution, even if its current second iteration. So, oops, let me just make sure I can forward my slides. Okay, so what I'm going to tell you about today really rides back on or is the follow-up. And again, it's a work in progress. I haven't spoken about this previously, but I thought this would be a great sort of occasion to present some of these new data. So what we've done previously is generate a single cell transcriptomic atlas of all endoderm from its appearance very early in development in what's called the glass stage, all the way through to mid-gestation when the endoderm comprises a simple epithelial tube that runs the length of the embryo. This is referred to as the gut tube. What I think distinguished what we did in terms of our single-cell transcriptomic efforts is that we focused just on the endoderm. And actually what we did was we microdissected endodermal tissue. If you think about this mid-gestational embryo in the gut tube actually comprises less than 5%, probably more like 3% of all the cells of the embryo. So if you were to take an entire embryo, as many have done, and associate it with single cells, the representation of endoderm would be very low. The endoderm comprises about 10,000 cells at this stage. So if you, for example, want 3 times overrepresentation, 30,000 cells, as I'm going to show you that we now have, you'd have to sample about 500,000 cells if you're going to do the whole embryo itself. And that, I think, is actually rather costly. So effectively, what we've done is we enriched through all stages through to mid-gestation. And a number of things came out of the study, which was essentially how does the endoderm of the mid-gestational embryo relate to the endodermal organs of the adult? And there were 2 major conclusions shown here. Firstly, as Rupa alluded to, sort of corroborating previous imaging data, we showed that the endoderm at mid-gestation comprises cells of 2 developmental origins. And I'll talk a little bit about that. And secondly, to our surprise, at mid-gestation, when the tissue is a tube, prior to the morphological appearance of any endodermal organ, we can see that it's patterned along the anterior-posterior axis, or AP pseudospace, predicted to cluster into cells that are transcriptionally similar, and each of these is similar to the endodermal organs of the adult. So you're getting this pattern even before the organs have started to appear along the entire gut tube. And this is actually a tube that is actually open in the middle, is closed at either end, and you can see domains along the anterior-posterior sort of axis that represent to the organs. And we can talk about this sort of, later. So what I want to do today is to sort of touch upon, again, work in progress, experiments where we're trying to validate and to chase later in development and into adults these cells of 2 developmental origins, to ask, well, at mid-gestation, the tissue is a mosaic. Is that the case later? Does that mean that our endodermal organs are mosaic organs comprising cells of 2 developmental origins? And then ultimately, with respect to the second I think, take home from the previous study, is that this patterning of the gut tube, and ultimately we're of course, performing more single cell experiments going later in time. But I think in thinking about tissue and morphogenesis, how organs arise in 3D space and over time, I don't think it really suits us to just be plotting things in pseudospace. What we want to do is to plot things in 3-dimensional space. And I'm going to show some of the data where now we have a fully annotated 3-dimensional gut tube. We can plot single cell transcriptomes, and we also now can plot epigenetics, so single cell attack data. Oh, okay. Something is happening strange here, but okay. So just in terms of this dual developmental origin of the mid-gestational gut tube, these are 30,000 single cells that we isolated from microdissected gut tubes. We processed them through the 10x Genomics single cell Chromium platform, and we showed, I think, conclusively by transcriptomics, but earlier we'd shown this by sort of imaging, that the gut tube comprises cells of 2 distinct developmental origins, embryonic and the so-called extraembryonic cells. We showed that in this sort of force-directed layout, we could cluster cells using one of the usual algorithms, and the clusters represented organ-specific identities, and these could be mapped in AP pseudospace. What was interesting was that cells, irrespective of their developmental origin, had a 99% correlation of gene expression at these stages, meaning cells of distinct origins would come together and actually look alike. However, we did identify a few genes that are more robustly expressed by these extraembryonic cells, and these are shown here. There's actually about 5 genes. What we're now doing is we're using mouse models to target these genes so that we can perform pulse chase experiments. So we pulse these extraembryonic or visceral endoderm cells early, chase them late, and ask the question, do later sort of endodermal tissues, do adult endodermal organs comprise extraembryonic cells? Because this would argue that by contrast to the textbook view of development, there's actually sort of two populations that will give rise to sort of our endodermal organs. Again, this is a work in progress, but I'll show you how we do this. So this is a prediction of this ROX5 in silico expression, and ROX5 is one of the 5 genes that seems to be upregulated in these visceral endoderm cells. What we would often do, or Sonia and Beth in the lab would do, is this is a computational prediction. This is a wet lab experiment. This is a microdissection of tissue out of an embryo where we can perform a whole mRNA in situ hybridization. I'm showing you this here because this is what we would be doing circa 2018, 2019. And I'm going to show you that we now have a technique that supersedes all of this. The thing about this is the endoderm is internal in the embryo, and to visualize it is very difficult because it's a simple epithelium and it's deep and it's kind of obscured by sort of masses of tissue that are sort of surrounding it. So we microdissect to look at sort of gene expression. What we often do is once we've validated the expression of a gene, we can use sort of CRISPR editing in embryonic stem cells to generate mouse models. And then we can recapitulate gene expression with reporters or transcriptional reporters. And this is an HPV reporter knocked into the ROX5 locus. In ES cells, we've made a mouse, and you can see this recapitulates the distribution of these cells chromogenically stained here. And this is just the GFP. So we perform these types of experiments to then generate an inducible Cre recombinase so that then we can perform pulse chase experiments.
Sorry, you stopped sharing your screen on the Zoom.
Oh, I did? Oh, I apologize.
No problem.
Do we have to get back out?
Yeah, we have to go to the Zoom, I think. There we go.
Take that photo too.
Oh, of course I took that photo. or every photo that is either I took it or someone in my lab took it. I'm glad you appreciate them. Tell members of my lab because they feel that I always set the bar high for images. Okay, so this is just a maximum intensity projection of a confocal Z-stat showing or where we've labeled extraembryonic cells with this ROX5 sort of a transcriptional reporter with a GFP. It's a nuclear GFP, and you can see the distribution is similar to an mRNA in situ. These experiments precede this experiment, which is where effectively we want to generate Cre-RT2s with various genes that are specifically expressed in these extraembryonic cells. And what we want to do is induce early chase late. But there's an issue here in terms of imaging these. I'm going to show you some later analysis. We're also looking in adults. But one of the issues we have is that this extraembryonic tissue will contribute about 20% of cells to the entire gut tube. These induction experiments are very inefficient. We label about 20% of cells. We're actually inducing around sort of very early post-implantation where we can't hit mothers with very high doses of tamoxifen, otherwise then abort their embryos. We can't sequentially administer tamoxifen because we want to induce early chase late and not have any cross-contamination with these embryonic epiblast-derived definitive endoderm cells. So the issue that we had in the lab, and people sort of hated me in my lab for this, was that we'd have to take serial sections and look for needles in haystacks. And people in my lab said, well, this isn't really an efficient way of doing experiments. We have to look for an alternative method to visualize these descendants when we're following them. And what we do is, this is the problem. You're looking for cells buried deep in our tissue. And what we now are using is a modified version of this IDISCO protocol for clearing. It's a protocol first developed in Mark Tessier-Levine's lab, published in 2014. It's been sort of updated by Zuhua Wu, who's actually a colleague at Cornell. These are the general steps. This is an embryo, and effectively what it does, it will allow you to visualize structures that are internal, which I think is really beautiful because you can preserve three-dimensional tissue structure, selectively label tissues that are internal and visualize them in 3D. So in terms of these pulse chase experiments that we're currently carrying out, and I'm only going to show you one data set, but I think it's really very provocative and I'm really excited. What we can do here is we can label the entire endoderm with cyan, and this is an antibody to EpCAM, and then we can visualize descendants of this extraembryonic endoderm in pink. So what's going to happen here is this is a 3D reconstruction of a light sheet microscope imaged specimen. It will spin. You will see the entire endoderm that is internal and is labeled. And then as it rotates a second time, you'll see some pink cells. There's not many pink cells, but there are pink cells. And I'm hoping that I can convince you that the pink cells, which are extraembryonic descendants, persist. Again, we're not labeling the entire population because our labeling is inefficient. And I'm also hoping to convince you that they are restricted to the endoderm and predominantly posteriorly. Okay, so you can see this, and I think this is beautiful 3D. And you can see these individual cells. You can see there is single cell epithelium, these are a columnar epithelium, and you can, I hope, appreciate that they reside within the endoderm specifically. So the alternative 2-3 years ago would be that people in the lab would have to cut millions of sections, they would complain all day, and it really wasn't effective. Now, armed with clearing methods and a light sheet microscope where a 5 to 8 hour image data acquisition can be reduced to less than an hour, we can actually be effective and sort of efficient in generating these data. Moving forward, what we're now doing is cataloging the distribution of these cells into later stages and in adult organs. But so far, what appears to be the case is that these cells do persist, arguing that our endodermal organs, or at least the hindgut and midgut derived organs and some of the posterior foregut derived organs, so sort of a pancreas, liver, and intestines, do comprise cells of two distinct developmental origins, embryonic and extraembryonic. Okay, so what I want to do now for the rest of my talk is to discuss, can we sort of take these type of force-directed layout that then we can bin and cross AP pseudospace, can we build a fully resolved 3D canonical gut tube where we can actually annotate spatially single-cell transcriptomic data? And can we actually input onto that as well single-cell attack data, so that we can truly sort of visualize, the transcriptomics that Rupert was talking about in 3D, in tissue context, which I think is where we want to go with our sort of experiments. And this is the work of Evan Bardo and Mo Gashi in my lab. And as Rupert sort of alluded to, this is an ongoing collaboration with Dana Pear's lab. Dana Pear is a computational biologist. She's never held a pipette. And so we work very well together. The project was spearheaded on Dana's side by Esther Werthoff, and Esther is a computational biologist. She was recruited to Alshos Labs at a salary higher than mine and Dana's combined. So she no longer works on the project. Josh Hess has taken over. And then Doran Haviv, I will show you, has developed a really critical algorithm that we're using to make this analysis possible. So the big questions are, how does the gut tube, of course, patterning set the stage for organogenesis? And the idea being, can we map single cell transcriptomes and chromatin accessibility onto 3D space and ultimately over time? Okay. So what I want to just talk about, so this is essentially sort of the idea. We're looking at this mid-gestational stage, and this is sort of computationally imaging in 3D, and then computationally segmenting the endoderm and also the kidney over time to understand how this structure sort of precedes the sort of formation of these sort of distinct domains in sort of 3D space. And you can see here a stomach, pancreas, lungs, and we know that this is patterned into these domains. I also wanted to just kind of touch upon this, again, the idea of why we need to just to understand, how development proceeds or any biological process proceeds in 3D over time. I mentioned how we can generate single cell data, we can map them, in pseudospace. And this is essentially anterior versus posterior. And this is just my schematic of a tube. But what we know about this tube is that there are two additional sort of orthogonal axes. There's a dorsal-ventral axis and a right-left axis. In terms of dorsal-ventral, when we looked at our single-cell data, we saw two clusters, which we label lung 1 and lung 2, that we now know are trachea and esophagus, and also pancreas 1 and pancreas 2, because the pancreas arises in a dorsal domain and a ventral domain. They then veer to the left, and then they combine to generate a single organ. So we know even at these stages, we're seeing clusters that are slightly distinct. And we just bundle them together, but we believe that they actually are sort of identifying sort of distinct spatial domains. The issue regards left-right patterning. Okay. So we know this is actually trachea and esophagus, and this is ventral and dorsal pancreas. Oh, and this is just to show you, I'm going to tell you how we do this imaging. PDX1 is a canonical pancreas marker. This is an whole mRNA processed the way we do in 2023. And I'm going to tell you how we do this, which is a three-dimensional reconstruction of the gut tube shown here in blue, and a heat map of PDX1 expression. You can see it in two domains. This is the dorsal pancreas, this is the ventral pancreas. And this really is a sort of benchmarking of these predictions, which are computational from single-cell transcriptomic data. Of course, single-cell transcriptomic data being sort of generated through the dissociation of tissues, where you lose spatial information. The other point I wanted to make is, I'm hoping I can convince you that there is dorsiventral patterning within this tissue at this stage. The issue of left-right patterning is still open. We know that the liver, for example, comes out in a bud that's ventral, but it actually veers to the right. As I mentioned, the dorsal and ventral pancreas veer to the left. The lung also comes out ventrally, but it's a little bit asymmetric, because I think I may have a movie to show you. And it also buds asymmetrically. And I think a long-standing question is, and I'm going to show you where we are. I'm not saying we have an answer yet, but this is something we're sort of moving towards. Is there left-right asymmetry in the endoderm or intrinsic to the endoderm, or is any asymmetric budding of organs sort of kind of instructed by neighboring tissues, namely the mesoderm, because the mesoderm sits right next to the endoderm. And we know that there's left-right patterning within the endoderm. We also know that there's left-right patterning within the endodermal lake. We don't know whether nothing has been reported early, but I think we're in a position where we can, where we should be able to identify genes that are left-right patterned in their expression at these very early stages. And I should actually just point out, when we're looking at this 8.75 stage, this is far earlier than most people look for the expression of these genes and the sort of the effects of these genes. So, for example, loss of PDX1 results in a complete loss of pancreas. But these mutants have only been reported and analyzed at least 48 hours after this stage. So this is 10.5 days onwards. So we're looking very, very early, and we're already seeing patterning. Okay, so this is just a foray. I put it into the lung organogenesis. These are, again, full mount image cleared specimens where we can visualize the lung in 3D. And I just wanted to show you that we can track lung development, again, in channel of a tissue of an embryo. And then if we look at this stage, so this is where we're actually doing all of our analyses. And here is, I'm going to show a high resolution rotating 3D view of this stage, where you'll see a clear asymmetry. And the question is the asymmetry we see here morphologically set up here molecularly? And this is just the whole embryo, and this is a close-up view. And I hope you can see that this is truly asymmetric. That the bud on the left side is larger than the right, and the budding actually veers a little bit, is not right in the midline. Okay. So in order to do this, so this just shows what we want to do. We want to take sort of our single cell transcriptomic data that we and everyone in this room probably has these kinds of data, these dimensionality reduction type plots. And ultimately, can we take this data and annotate it in 3D? So we can sort of generate, 3D data either by antibody staining. I'm going to now show you a protocol for mRNA in situ that has really been a game changer for us in the mouse that we're now using. And ultimately, what we want to do is to generate a fully annotated gut tube. And here what we're doing is, this is in voxels. Every voxel has been approximated to the size of a cell, recognizing that this structure comprises about 10,000 cells at this stage. So what I wanted to just quickly sort of remind you of what we've done in the past. As I said, we'd enrich for the endoderm, single cell dissociation, load them onto the 10x Chromium platform and analyze. These are the folks in my lab. These are sort of Dana's folks, and we work really well together given our sort of complementary expertise. So the idea of what we've done before is we can make, or Dana and Manu can make predictions about genes expressed along anterior posterior pseudospace. We were particularly interested in transcription factors that we thought really sort of defined distinct identities of tissue along pseudospace. And then we validate them with these, as I showed you previously, microdissected gut tubes, and we do chromogenic stains. And chromogenic mRNA in situ have really been the sort of benchmark in situ protocol in the field for about 30 years. And really, they're not particularly, they serve a purpose so that you can actually see expression of genes within the gut tube, but they really aren't sort of ideal. So this is what we can do today. So these are gut tubes where we can look at gene expression, and these are genes that are characteristic of different organs, and we can visualize them in 3D and quantitatively, and I'm going to show you how we can do this. This is mainly the work of Mo Gashi, and the protocol, which is this hybridization chain reaction protocol. I actually should I should admit that I went to a meeting at the EMBL in 2019, and Mark Lewandowski, who I think Janine Wellick knows very well, was there. And Matt Anderson, who's in Mark's lab, gave a talk. And I have to say, he showed some HCRs of genes expressed within the mesoderm. And it was one of the first times that I literally fell off my chair. Because every couple of years, there'll be a paper showing a proof of principle gene expression pattern and a fluorescent in situ of a mouse embryo. Only sort of genes with good signal to noise will work. It doesn't work for 80% of your favorite genes. This protocol, we've been using it for about 2 years, works for over 85% of any gene we like to try, which has really been, I have to say, a game changer for us. I'm not going to go into details. HCR has been developed in Niles Pierce's lab at Caltech. There have been multiple iterations that have been published in a series of manuscripts. What is nice is it will allow you to visualize several probes, up to 5 currently. So it isn't as multiplex as some of the sort of section in situ, like Murfish or Seepfish, where you can do hundreds, but it gives you 3-dimensionality. And I'm going to show you how we're using this approach and combining it with imputation of single cell data so that we don't need to perform Murfish types approaches. We can quantify fluorescence if we want. We can look at single molecule in tissue context deep inside an embryo. The protocol for me is quite short. My lab would say, well, this is 2 to 3 days. This is a long protocol. And I'll tell people that when you do a chromogenic in situ, it's about 5 days. So this is actually, I think, high speed in situ. The only downside is the probe design is performed or is done by Molecular Instruments, which is a company set up by Niles Pierce. So you don't have much control over probe design, but you can work with the company to, for example, generate probes that will detect specific isoforms of genes of interest. The other thing we do for our HCRs is that we clear, and we use the C3D protocol, which is distinct from the IDISTO. It just happens to work better for these types of samples. This is, this was developed, I believe, by investigators at the NIH around 2019. So we use different types of clearing protocols depending on the experiment. Okay, So HCR works, I would say, beautifully for very early sort of embryos, pre-implantation, which is the stage my lab are also interested in, but also post-implantation. And here we're looking at three genes and DAPI. We use DAPI, or we have in the past used DAPI to stage embryos, so we can go up to 5-color imaging. This is a maximum intensity projection. And effectively, we built a pipeline to perform analyses. So we take a Z stack on a laser point scanning confocal, but as I was discussing earlier today with one of my meetings, we're also now testing the light sheet. I'm not sure whether it will give us the resolution, but when the advantage of the light sheet is we can increase the speed of the image data acquisition. We use a confocal with a very large field of view, so we can capture an entire embryo with a single sort of 10x objective. What we then do, so that's kind of what it looks like, and effectively what we do is we select a number of genes, one of which, Claudin 6, so we now have up to 5 genes, will be expressed throughout the entire gut tube. The importance of this is that we can use this gene, which is expressed throughout the gut tube, but also elsewhere, for example, in the face, but we can go in and manually segment the gut tube based on this gene expression. So this is Claudin 6. We can then 3D reconstruct and create a mask of only the gut tube, and then we can sort of plot onto that, you know, the Claudin expression cleaned up so that we've segmented only the gut tube and then projected it onto the DAPI channel. So we don't have all the expression of Claudin outside of the gut tube. What this allows us to do is to then go in when we're looking at multiple probes and segment only the gut tube so that we can extract details of the expression of any gene of interest within our target structure. So this is a 3-dimensional image data segmentation that will allow us to focus in on the tissue of our interest. So rather than micro dissect, what we now do is generate 3D image data and then use a computer to isolate the tissue of interest, and then we can project expression sort of domains. And then we can play around with things, and these are the same kind of genes plotted here along AP pseudospace. You can see thyroid and liver, you know, for hex, and then we can also plot heat maps. Okay, so in considering how to set up this project, we talked a lot with our computational colleagues, and we made them aware that individual markers will reveal different distributions and capture different regions of the gut tube. What they came up with, and they said, well, you know, what we'd like you to do is, can you look at, and they ballparked it. Originally, it was 30 genes that would have so they went into the single cell data, and they looked at that, and they could make predictions. I'll show you on the next slide. And they said, well, we think, well, originally they said, we think 30 genes should be sufficient. Look at 30 genes in different combinations, and then in these types of HCR data, you know, segment the gut tubes, give us the data, and let's see what we can do. 2 years later, we've now looked at 70 genes. So we're kind of improving, you'll see our resolution. And it's, you know, again, it's been a constant discussion with our computational colleagues who want a lot of data. So you tell them 30 and then they want 60. And then you tell them, you know, you have 100 embryos, and then they say, well, we want, you know, 200. And then when they say 200 images, and we're like, no, each image is about 150 images because they're 3-dimensional. So you're working with 25,000 images. So this has been an ongoing conversation. So this is just to show you that they're using various methods for anyone that's computationally inclined here, such as this metric Moran's eye. It's been applied in Drosophila to construct a 3D spatial transcriptomic atlas of Drosophila a couple of years ago now. And this was a study carried out in Nikolaus Strajewski's lab in collaboration in the Max Gelbuch Center in Berlin, in collaboration with Nir Friedman at the Weizmann Institute in Israel. So they can use this Moran's eye to look for genes with high spatial autocorrelation. They can make predictions, and then we can ultimately go in, do experiments and validate their predictions, and we can perform. This is just actually progress to date is we have now performed 250 data sets, and we've analyzed 72 genes in multiple combinations. So we have Claudin 6 or an equivalent to always delineate the gut tube, and then we look at 3 or 4 additional genes in various combinations. So in order to construct an atlas, I think we realized very early on is that even if we took embryos that were very carefully stage matched, so these are 13 to 15 somites, what we call mid-gestational embryonic date 8.75, they've all been stained for these, 3 genes, DAPI and Claudin 6, the pan-gut tube marker. You'll notice they vary a little bit. So even replicates exhibit morphological variations. So what we discussed with our computational collaborators is, could we take these replicates where we've done our which we put through our image segmentation pipeline and generate a canonical duct tube structure, and ultimately average everything out, sort of a common coordinate framework, so to speak, as is being done for, I think, the Allen Brain Atlas of generating a common coordinate framework for the brain, or the mouse brain, and probably the adult human brain. So this is just 3D reconstructions of the tissue, heat maps of this one gene, and you can see there's slight variations. So effectively what Dana Pear's folks did, and this is started by Esther, as I mentioned, followed up by Josh, was that they've effectively sort of performed various analyses. Again, I will sort of show you these slides, but these were experiments or analyses performed by our computational colleagues. I know what they did. I can't vouch for every detail of the computation, because so I plead ignorance, because I'm the biologist in the collaboration. So effectively what they're doing is, what they've done with our data is to generate a CCF, or common coordinate framework, a kind of generic gut tube structure. And ultimately, in the interest of time, I'm not going to go into the details, but we're looking at things like where the liver is to figure out whether we can use that as a midpoint reference to readjust the atlas. But this is essentially the idea. This is our 72 marker 215 Z-stat containing reference atlas, and it is 3D. And what is projected here is the 3D average across multiple replicates expression of one gene Ripley3. So the question then becomes, well, we've done this for our 72 genes, but how can we go genome wide? And I think this is the important part. Well, we can keep doing genes up the wazoo, but I don't think I could convince anyone in my lab to do over 2,000 genes, which is what the number of genes expressed in the gut tube. I also don't think my lab have the finances to do those experiments. So if one can construct from a minimal set, in this case 72 genes, a reference atlas, how can we then impute genome-wide our single-cell RNA-seq data, which is over 2,000 genes? And can we also then impute epigenetic data? So that this is, I think, where we're sort of starting to go biologically. This is I think, the current, I would say, best example of a spatial transcriptomic atlas of a mid-gestational mouse embryo. It was published about 6 months ago. We were peripherally involved in this study because they used our endoderm data set and combined it with a whole embryo single cell data set, but also we performed some experiments validating some of the endoderm sort of markers shown here. Effectively, what I'm showing this for is that this individual, Doron Haviv, in Dana's lab, has recently developed what is referred to as a variational autoencoder. Do not ask me how it works. What I can tell you is what it does is it will impute single cell data onto spatial data. He's writing up his first paper, and what he's done here is he's compared his autoencoder for imputational of single cell data onto spatial data, and he's benchmarked it against some of the more sort of, I guess, the very popular imputation methods, such as NovoSpark or GIMV, and then some of the Tangram. And this is sort of the sort of the true data. This is fish data. So this is kind of this is experiment ground truth, and these are imputation. And envy, from what I see and what my computational colleagues tell me, seems to outperform many, if not all, of the current imputation methods. Doron and Dana are writing up envy. It will appear on bioarchive in the next couple of weeks for anyone who's interested in sort of looking at this. What this is doing is imputing single cell data, but it's two-dimensional. So what Doron has now done for our project, having benchmarked envy and shown that it outperforms any of the current imputation methods, is he has been able to perform imputation in 3 dimensions. I'm not going to, for the interest of time, and this is all sort of Dana's work, but effectively what he's done is impute over 2,000 genes from single cell RNA-seq data into the 3D sort of mean atlas. Okay, and we've also done multi-ohm, but I'm not really going to talk about that. So we actually have both accessibility and RNA-seq. So these are just some examples of a sort of reference data set where we have an HCR data, as I've shown you for HEX, and then a prediction down here for Wnt9b based on MB. These are the 3 axes, anterior, posterior, dorsal ventral, left, right, and I apologize, this is kind of, the resolution isn't great, but this is a 3D reconstruction, and this is 3D of Wnt9b, where you can sort of query any gene of interest on this 3D atlas. And this is a prediction. We never performed an HCR mRNA in situ hybridization for the construction of our atlas, but after they'd sort of, we'd run this sort of test run, we could then do an experiment, and then we're validating the computational prediction, and there is agreement. This is just, again, demonstrating that the imputation works, and this is where one can leave out individual genes, and the atlas still sort of is robust. I'm not going to go into the details. This, I think, is kind of, I'd say, very convincing to me. So this is the previous 2D organogenesis atlas. These are all the cell types. This is the endoderm removed out of this atlas. So the endoderm, as I mentioned, is here in pink. This is our 3D atlas. And so these are 2 genes, Wnt5a and ILOT1. And ultimately, I hope what I can convince you, or this can convince you of, is that our 3D atlas agrees and improves the 2D atlas. And then we can make predictions for left, right, and dorsiventral. This is the current accuracy. We're very accurate in the AP, of course. The dorsiventral, I think, is acceptable. The left, right, we're working on, and we have a couple of tricks regards how we're going to improve these numbers. So we can really start to really sort of interrogate this atlas that is, to my knowledge, the first 3-dimensional single-cell RNA annotatic atlas. Okay, in the interest of time, I'm just going to go on to where we're taking things. And again, this is a work in progress. I'm going to tell you where where we're excited to move things forward. So one of the things that we're very interested in is how this anterior posterior sort of pattern is set up. What we thought was very interesting when we sort of plotted this transcription factor data, and I think this sort of, I would love to discuss this with Janine Wellick. If you look at Hox genes, and Hox genes being the general sort of coordinate system that the embryo uses for sort of identifying sort of axial sort of, or for specifying sort of, or reading out axial identity, we have a Hox B1 over here in the middle and a number of Hox genes clustered posteriorly. But we don't have a nice Hox code that runs from anterior posterior at this time. In the mesoderm, there is a Hox code, and I think Janine Wellick would be sort of the expert here. But as far as I've read the literature and seen data, there is a Hox code that runs anterior through posterior at this stage within the mesoderm. So this argues how ultimately is this AP pattern set up, which is something we're very interested in. We know that later the Hox code is elaborated anteriorly, but early on it appears not to be. We also know from existing literature and previous studies that there are particular genes such as CDX2 or HOXB1 or PRRX2 that seem to specify distinct organ identities. And these are from studies of knockout mice analyzed very late in development, or much later than the gestation. So what we're doing right now, and again, this is work in progress, is we're adopting, I spoke to a number of individuals about this Degron system that we've, shown works very efficiently in mouse embryos and also in pluripotent stem cells, where we can generate a Degron tag onto a protein of interest. And these are going through the pipeline right now. We have mice, we're breeding them. And ultimately, what the Degron tag allows us is the ability for acute perturbations and degradations of proteins. So we can go in and within 6 hours, we can get full degradation of any protein of interest, with the caveat we've only looked at transcription factors so far. So this allows us to go in and degrade, isolate gut tubes, and then perform single cell omics. We're also, we're performing perturbations, and we're starting with, as I mentioned, key transcription factors that seem to specify distinct organ identities. And also one of my favorite ones, SOC17, that specifies endoderm, because I had to throw this one in. And then we're also looking later regards developmental progression. We're taking it to begin with just 24 hours from E8.75 to 9.75, but we've now collected data from 12 hours later, and I can sort of show you, this is again a work in progress. This is what we had. This is single cell RNA-seq from 9.25, the entire gut tube shown here. And then we also have paired attack-seq data, so we can sort of, we know both the accessibility and the transcriptome. This is what we're currently working on. It's very much, as I said, a work in progress, but I can show you at least one tidbit that I think is rather interesting. So this is a gene called CBPA. It's a transcription factor. It's a well-known marker of the liver. And as you can see in the single cell RNA-seq data, it's specifically expressed in the liver. However, the single cell attack data is showing us that there's accessibility and peaks are open across the majority of the posterior of the embryo. And we think this is really very interesting. So this is something we're keen to follow up on, because we also know there is data from a number of labs performing perturbations, as we plan to do in the key transcription factors, that demonstrates, for example, that you can take out a transcription factor that specifies pancreas, but pancreas will readily change to a stomach type identity. So where does this intrinsic plasticity that's existing within the endoderm arising from? And I think a lot of the clues will come from perturbations and then pairing up single cell attack with single cell RNA-seq data and doing this in 3 dimensions. Okay, and then I just want to finish on this idea that, where is the patterning coming from in terms of sort of generating distinct transcriptional identities of organ-specific clusters of cells across the anterior posterior axis. This, where does the patterning come from that can be sort of mapped in sort of transcription factors that are uniquely expressed in domains along the AP axis. And I just want to highlight this experiment, which is us, I think, going through the garbage can, but I think we realize now that we may have discarded something that was very interesting. So when we first performed our single cell RNA-seq experiments, as I mentioned, we've microdissected. And everyone in the lab involved in the microdissections, they'd also get me to help, because it was really a group effort. It needed a lot of people to be isolating gut tubes and dissociating. It really annoyed us that we could never isolate a clean gut tube. There was always mesoderm next to it. And we tried very hard to pick off mesoderm, and then we damaged the gut tube, and then we thought, well, we don't want to do that because the transcriptomes might not be great. And then we realized that perhaps this may not be a bad thing, because what we're actually isolating or taking with the gut tube is the mesoderm that sits adjacent to it, 2-3 cell diameters next to it. It's not the mesoderm that's 10 cell diameters away. It's the stuff that encompasses kind of the entire gut tube. And it's probably the cells that are performing crosstalk and might be patterning. So we never went in our first study to look at the mesoderm. We threw it all out and said, this is not interesting. We don't work on mesoderm. And now we realize we need to go back and start to analyze this mesoderm. So for example, we isolated 30,000 cells of the endoderm, which I told you about. Well, we actually have an equivalent number of cells of the mesoderm. So we actually have all of this in our data, and we're starting to mine it. And this is really my last slide, but effectively, this is again a force-directed layout where we plotted the endoderm over here across AP pseudospace, and then the mesoderm, and we know who's next to who. We can make predictions regarding signaling, receptors and ligands in mesoderm versus endoderm, and we can test them experimentally. And there's also, I think, something interesting. I keep telling my lab, and then our computational biologists say, well, you need to isolate more cells, which we may do. But we also see cells that seem to be somewhere transitioning between mesoderm and endoderm. And again, arguing that perhaps in this sort of pharyngeal region, which is where the neck is, there may be sort of cells crossing over from one lineage to the other. The direction for which we don't know, we have to do a fake mapping experiment the way we're doing for this extraembryonic population. But I think this is telling us many things that seem to be very interesting that we will be following up on down the line. Of course, now that we also have mesoderm, we should be able in the future to map the mesoderm transcriptomes at single cell resolution adjacent to the endoderm, the way we've done for our 3D canonical spatially resolved endoderm atlas. Okay, that's my last slide. I'm going to stop there. And as I alluded to, this is the work of a number of people, past and present from my lab. Mo's done a lot of the single cell spatial transcriptomic project. Sonia and Beth are performing the fate mapping and doing the sort of sample or tissue clearing to chase these extraembryonic cells. There's some former lab members. Evan Bardo was involved in setting up the HCR. UW-Madison. I didn't talk about his work, but he really was the person who established the Degron approach that Mo, Sonia and Beth are using, and others in the lab. All of the single cell genomics projects in my lab, and also many of the imaging projects, are a collaboration with my colleague Dana Pear, who's a computational biologist. And these are the folks in her lab that have worked with us over the years. And I would be remiss if I didn't thank Mark Lewandowski, because it was really his lab that presented this 3-dimensional whole mountain situ hybridization that is robust, reproducible, and really works in mouse embryos, that really got us going on the spatial transcriptomic project. And I have to say he's been great, because Matt Anderson hosted Mo Gatti to their lab and showed him the protocol for mesoderm, and we've now adapted it for looking deep in the embryo and at the endoderm. I'm going to stop there. Thank you for listening, and I'm happy to discuss further.
Thank you, Jerry. I don't know if I will quote that correctly, but when you look at the mesoderm and expression of ligand and receptor, it's like that both of the mesoderm and the express both ligand and
it depends. Those are kind of cherry picked. So it depends. This is, oh, sorry, I'll just go back. These were just cherry picked. Oops, let me go back. See what I can do here. I think this is, to be honest, there's it really depends. So it's you're right that, for example, Win3A is posterior in both. Frizzled, for example, GIF1 is not. So these are is not. So these are just cherry-picked examples.
I was wondering if there might be the fourth one here.
Then absolutely, I think you're probably correct. And depending on which ligand receptor combination we're looking at, there probably is. This is just a gold mine, and I think the goal will be to focus because there's a lot of questions to ask. Question. So what I didn't appreciate until I thought about it recently was that when you think about the gut tube, you think about the foregut, it really undergoes two very distinct behaviors. Within the foregut, the organs will bud and then they will undergo organotypic specific branching. You know, the pancreas branches differently from the lung, branches differently from the liver. Mid and hindgut intestine don't actually bud out, they then loop. And you know, I think there are mechanisms that have been put forward by Cliff Tabin, by Natasha Kurpios, who used to be with Cliff Tabin, regards what might be driving looping of the intestine, but far later. I'd say at least 4 days later in the mouse embryo. In terms of this dorsoventral, I think this is really interesting, and it's really exemplified by the I can actually just, if I go back to here, where is it? Here. So as I mentioned, we said we had identified these two lung clusters that we call lung 1 and lung 2 originally, and then when these guys did their spatial transcriptomics, their 2D spatial transcriptomics, we could then, you know, our contribution, I think, to the study was to show that our lung 1 cluster over here was in fact esophagus, and this, or was trachea, and this was esophagus. The idea being that when you make a lung, and again, I think Janine probably understands this better than I do, you have an original branch of the bud that then will asymmetrically branch left and right. That will then zip up and separate into the 2 tubes, starting where the lung will branch out. Let me just, I may actually have this. So effectively yeah. So effectively what you're seeing here is, if I don't know if it's clear, here you get the bud that will bifurcate. Over time, this will then zip up and zip up further. And this is your trachea, and behind it is the esophagus. The trachea ends with the lung, and the esophagus ends in the stomach. And I think what we're seeing in terms of dorsal ventral patterning at this very early stage, before any branching, is patterning that will give rise to this behavior that then causes this successive chain of events to elaborate into these structures. So from 1 tube, you're making 2 tubes, and your elaboration branching organs. And what is, I think, very interesting is at the, where are we? Yeah. Is at the, you know, there's this very elaborate branching here, thymus, thyroid over here, but here you're just getting looping. Okay, so you should be answering these questions. You probably understand the lung better than I do.
No.
Right. Let me do that. Absolutely. Stage, there is a distinct pattern of hot gene expression. The question we want to ask is, given that the endoderm shows asymmetric branching And is there any intrinsic asymmetry within the endoderm, or is any asymmetric branching constructed by the mesoderm? I think that we have a couple of candidates regards left-right from our transcriptomic atlas. The atlas, as I mentioned, is not as accurate as we would like it to be. We're working to improve it in terms of, I think, just setting the midline correctly, because this thing skews a little bit. And then we'll be able to answer that question. It may all be in the mesoderm, but there is no data in the literature right now, either in mouse, in chick, or in xenopus, that argues one way or another for left-right asymmetric gene expression within the endoderm. I think the only data comes from xenopus, and it's Mike Levin that has shown some asymmetric, I guess, not gene expression. I think he has electric currents or something. And Mike Levin has some unusual hypotheses regarding left-right asymmetry. But to my knowledge, nothing has been shown within the endoderm. And we know the endoderm is a left-right asymmetrically patterned tissue. It could all come from the mesoderm. So that's why we want to look at the mesoderm that's right next door.
Can I ask a provocative question?
Yeah.
So, I was very struck by your PDX2, where you can actually see that the PDX2 expression and, the specifying early on before the PDX2 knockout dies, which is
Yes.
later,
Yes.
right? And taking that with your CEBP data at the
Yes.
end, is that, when the epigenome completely shuts and you don't have everything else open elsewhere, that's when the knockout of the transcription happens.
It could be. So that's what we're taking. We're doing multi-ohm single cell attack and single cell RNA to later stages. But what we think is very interesting is not that the chromatin is not. There seems to be some kind of code as to where it's open, and that's where we're hypothesizing that could it be that we can mutate genes within a particular region and actually get reprogramming, or and outside of that region we don't. And that seems to be, and there's something unusual with the lung, liver, pancreas region.
And and maybe it's not chromatin at all. I can't believe I'm saying that in public, but no, I'm also wondering whether at some point, and I don't know whether the neural crest somehow contributes to the endoderm.
No, it doesn't.
Oh, okay.
No, that.
So I was wondering whether that compensates, and at some point it just stops.
No.
Are there questions in the chat?
No, I was just no, it was
Okay, all right.
Yes. So what we did was,
Just about the,
yeah. So what we did was, I didn't go into the details. We did bulk of whole gut tubes. We did bulk of gut tubes. We bisected into 4 quadrants. We did pseudo bulk by aggregation with single cell. So when we first saw this kind of major, I guess the principal component was the spatial component along AP, we didn't believe it. So then we did bulk micro dissected into quadrants to see whether we could somehow order our clusters, and we could. So we've done bulk for every single cell experiment, we've done many bulks. Nothing. So what was interesting is if we pseudo bulk the single cell and we compare it to the true bulk RNA-seq, nothing distinct came up with the bulk RNA-seq. Oh,
The light sheet that you have, like,
yes, it does. So we use a number of light sheets. We use them the light sheets that we use are commercial, we don't build our own. They're marketed by Brooker in the United States, and they're based on an instrument developed by Jan Ellenberg at the EMBL. He created a company called Luxendo. Luxendo is marketed by Brooker over here. We use what they, their movie spin, which again has a multi-view capability, and then for very small embryos, and I didn't show you any today, where we time lapse, we use their inverted, which is their true live. So we have different setups depending on the sample. So large specimens that are fixed and cleared, we have the multi-view movie.
How many views?
It depends. You can set it up different ways.
Like how,
Huh?
like how many views?
It varies on it also varies on how much usually I think it varies because the sample rotates and you can have it yeah. I think one of the I couldn't tell you off the top of my head how many views for this one. All I know is that the data sets are huge. And whenever doing any of these experiments, what we've now realized is it's not just the acquisition of the data, it's being able to move the data, process the data, and work with the data. And that's kind of a major investment.
Yeah.
Well, I wouldn't say it was supercomputer because these days I think that's a standard computer, but it's a workstation with major imaging sort of capabilities, GPUs and so forth.
Right, if there aren't any more questions. Oh, sorry.
Yeah. That's a very interesting question, which I get asked all the time. What would happen if you deleted them? Again, we haven't done the experiment. In principle, we could do it with a diphtheria toxin allele, but again, our ROSA, or not our ROSA, our 3RT2 is not very efficient. So we never hit everything. But I would stick my neck out and say, you could delete them and nothing happens. But so the question is, why do you have them there? The reason I think you have them there is because the endoderm forms through a very unusual morphogenetic behavior, which is this widespread intercalation, where two cell types will intercalate, and we think that the morphology of the embryo dictates that it has to form this way. So you end up having this mosaic tissue. The question, I think, the important question to me is not whether if you delete it, can you make a normal gut tube, or can you survive? It's if you have a disease or a disease model that hits one or the other. Say we activate an oncogenic driver in the intestine, in a visceral germ or an extraembryonic descendant or an embryonic descendant, do they behave differently? So I think that I suspect that the way we'll see a difference, or where we might see a difference, is when we challenge them. Because we've also done an experiment that we haven't published, a SOX17 mutant. It's one of my favorite genes, where SOX17 mutants don't make definitive endoderm, this embryonic endoderm. However, and we've not published this, SOX17 mutants will make a gut tube that patterns normally. It just isn't, the diameter isn't big enough to allow the embryo to survive for reasons that are unbeknown to us. But the tube actually patterns normally. So this argues that you can pattern the tube if you don't have the embryonic population there.
All right, it's fine.
Thank you.